In this lecture, we will discuss about the types of DNA polymerases, particularly prokaryotic polymerases, that is DNA polymerase I, DNA polymerase II, and DNA polymerase III. We will discuss their structures, functions of their subunits, and activities. So let us start our discussion on DNA polymerases with DNA polymerase I. Arthur Kornberg discovered the first E. coli DNA polymerase in 1958. Although Pol-1 is a single 102 kilodalton polypeptide chain, it is remarkably versatile. It catalyzes three distinct reactions. It has a 5' 3' polymerizing activity, and it also has two different exonucleases activities, a 3' 5' exonuclease activity and a 5' 3' exonuclease activity. This is the animation of the polymerizing activity of DNA polymerase, where it adds the incoming nucleotide to the growing strand. The question is, why does a DNA polymerase need two exonuclease activities? The 3' 5' activity is important in proofreading newly synthesized DNA. If Pol-1 has just added the wrong nucleotide to a growing DNA chain, this nucleotide will not base pair properly with its partner in the parental strand and should be removed. Accordingly, Pol-1 pauses and the 3' 5' exonuclease removes the mispaired nucleotide, allowing the replication to continue. This greatly increases the fidelity or accuracy of DNA synthesis. The 5' 3' exonuclease activity allows Pol-1 to degrade a strand ahead of the advancing polymerase so that it can remove and replace a strand all in one go of the polymerase, at least in vitro. This DNA degradation function is useful because Pol-1 seems to be involved primarily in DNA repair, for which destruction of the damaged or mispaired DNA, and also for RNA primers during primer removal and its replacement by the new DNA. We start with two adjacent progeny DNA fragments, the right hand one containing an RNA primer which is red in color at its 5' end. The two fragments are separated by a single-stranded break called a nick. DNA polymerase I binds to the double-stranded DNA at the nick. DNA polymerase I simultaneously removes the primer and fills in the resulting gap by extending the left-hand DNA fragment rightward. It leaves the degraded primer. DNA ligase seals the remaining nick by forming a phosphodiester bond between the left hand and the right hand progeny DNA fragments. It appears as if the nick is moving or translating, and that is why this activity is called the nick translation activity. Another important feature of DNA polymerase I is that it can be cleaved by mild proteolytic treatment into two polypeptides. A large fragment known as the Leno fragment. This fragment is made of 604 amino acids and has the 5' 3' polymerizing activity and the 3' 5' exonuclease proofreading activity. The other fragment is the small fragment which is made up of 323 amino acids and it has the 5' 3' exonuclease activity. The Leno fragment is frequently used in molecular biology when DNA synthesis is required and destruction of one of the parental DNA strands or primer is undesirable. Now the main question was, is DNA polymerase the main replicating enzyme? DNA polymerase I is encoded by the gene Pol A. What would happen to a Pol A mutant. In 1969, Paula DeLucia and John Keynes isolated a mutant with a defect in the Pol A gene which codes polymerase I. This mutant lacked polymerase I activity, yet it was viable, strongly suggesting that Pol I was not really the DNA replicating enzyme. Instead, Pol1 seems to play a dominant role in repair of the DNA damage. It fills in the gaps left when damaged DNA is removed. The finding that Pol1 is not essential spurred a renewed search for the real DNA replicase, and in 1971, Thomas Kornberg and Malcolm Gefter discovered two new polymerase activities, DNA polymerase II and DNA polymerase III. We will see that DNA pol 3 is the actual replicating enzyme. But before that, let us talk about the DNA polymerase II. DNA polymerase II is the second DNA polymerases to have been found in E. coli, and it plays an important role in DNA repair rather than DNA replication. It is encoded by the Pol B gene. It has 5' 3' polymerizing activity, and it also has the 3' 5' exonuclease activity for error correction. Polymerase II is not essential for DNA replication under normal conditions, but it is important for DNA repair. It acts as a backup enzyme during replication if DNA polymerase III stalls or encounters a lesion. Pol 2 has moderate processivity, that is, it can synthesize some DNA but not very long strands of DNA. Let us now talk about the DNA polymerase III, the main replicating enzyme. The enzyme that carries out the elongation of primers to make both the leading and the lagging strands of DNA is called the DNA polymerase III holoenzyme. The holoenzyme designation indicates that it is a multi-subunit enzyme. The holoenzyme contains 10 different polypeptides. On dilution, this holoenzyme dissociates into several different subassemblies as indicated in the table on your screen. It was found that three polypeptides compose the core of the polymerase III: the alpha, the epsilon, and the theta subunits. These have molecular masses of 130, 27.5, and 8.6 kilodaltons respectively. The rest of the subunits of the holoenzyme dissociated during purification, but the core subunits were bound tightly together. The alpha subunit of the polymerase III core has the DNA polymerizing activity. The core has a 3' 5' exonuclease activity that removes the mispaired bases as soon as they are incorporated, allowing the polymerase to proofread its work. The epsilon subunit has the 3' 5' exonuclease activity that carries out this proofreading. The function of the theta subunit is unknown. However, it is clear that the alpha and the epsilon subunits cooperate to boost each other's activities in the core polymerase. The Pol 3 core enzyme by itself is a very poor polymerase. It puts together about 10 nucleotides and then falls off the template. Then it has to spend about a minute reassociating with the template and the nascent DNA strand. This contrasts sharply with the situation in the cell where the replicating fork moves at a tremendous rate of almost 1000 nucleotides per second. Something was definitely missing from the core. That something is an agent that confers processivity on the holoenzyme, allowing it to remain engaged with the template while polymerizing at least 50,000 nucleotides before stopping. The holoenzyme owes its processivity to a sliding clamp that holds the enzyme on the template for a long time. The beta subunit of the holoenzyme, which is a ring-shaped structure, performs this sliding clamp function, but it cannot associate by itself, and hence it needs a clamp loader to help it to join the complex. And a group of subunits called the gamma complex provides this help. The gamma complex includes the gamma subunit, the delta subunit, the delta prime subunit, the xi subunit, and the phi subunit. The sliding clamp is a ring-shaped structure made of two monomers of beta subunits. The beta clamp can bind to DNA and slide over it. However, just discussed before, the beta clamp cannot get loaded onto the DNA template by itself. It rather requires a clamp loader, that is the gamma complex, to load it. The gamma complex is made up of 5 subunits as can be seen on the screen. First of all, ATP binds to the gamma complex, as a result of which the delta subunit of the gamma complex binds to one subunit of the beta clamp. Binding of delta to the beta monomer leads to a protein-protein interaction, leading to a conformational change in the beta monomer. This conformational change results in a change in the curvature of the beta monomer, leading to the ring opening. The beta clamp can now be loaded onto the DNA template. Hydrolysis of ATP takes place, driving delta subunit away from the beta monomer, resulting in the regaining back of the conformation and hence the curvature. This results in ring closure of the beta clamp. The end result is that the beta clamp is loaded onto the DNA template. Let us have a look at whatever we discussed just now. So, we have the sliding clamp with two subunits forming a ring. The clamp loader loads the sliding clamp onto the DNA template. One way we can imagine the beta subunit conferring the processivity on the DNA polymerase core is binding both the core complex and the DNA. That way, it would tie the core to the DNA and keep it there, hence the name beta clamp. Let us quickly revise the function of the gamma complex, the clamp loader. The gamma complex loads the beta dimer onto the template primer junction. Binding of the clamp onto the loader needs ATP. The next step is exchange of the loader from the clamp to the core complex. This needs ATP hydrolysis. At the replication fork, there is a DNA polymerase with two core enzymes, one for the leading strand synthesis and other for the lagging strand synthesis. However, these two core enzymes do not have an independent existence. They are dimerized to each other by a tau dimer. There are two beta sliding clamps for both core polymerases, but there is only one clamp loader to load these beta clamps, and this is associated with the lagging strand. Hence, the DNA polymerase III is called an asymmetric holoenzyme. This is the structure of the sliding DNA clamps. They encircle the newly replicated DNA produced by an associated DNA polymerase. The sliding clamp interacts with the part of the DNA polymerase that is closest to the newly synthesized DNA as it emerges from the DNA polymerase. How does the association with the DNA with the sliding clamp change the processivity of the DNA polymerase? In the absence of the sliding clamp, a DNA polymerase dissociates and diffuses away from the template DNA on average once every 20 to 100 base pairs synthesized. In the presence of the sliding clamp, the DNA polymerase still disengages its active site from the 3' OH end of the DNA frequently, but the association with the sliding clamp prevents the polymerase from falling off the DNA template. By keeping the DNA polymerase in close proximity to the DNA, the sliding clamp ensures that the DNA polymerase rapidly rebinds the same template primer junction, vastly increasing the processivity of the DNA polymerase. When the DNA polymerase reaches the end of a single-stranded DNA template, the presence of double-stranded DNA in its active site results in a change in the conformation that reduces the polymerase's affinity for the sliding clamp and for DNA. Thus, when a polymerase completes the replication of a stretch of DNA, it is released from the sliding clamp so that it can act on the new template primer junction. Thus, DNA polymerase III is a highly processive enzyme. The effect of processivity is quite remarkable. In the absence of the beta subunit, DNA polymerase III falls off the DNA template after a few dozen nucleotides have been polymerized. Its rate is 20 nucleotides per second approximately. In the presence of the beta subunit, DNA polymerase III stays on the DNA template long enough to polymerize up to 50,000 nucleotides. Its rate is approximately 750 to 1000 nucleotides per second. With this, we come to an end of this lecture. In this lecture, we have discussed the types of the DNA polymerases, particularly DNA polymerase I, DNA polymerase II, and DNA polymerase III. In the next lecture, we will talk about the other enzymes and accessory proteins required for DNA replication. Thank you.